tissues that deliver oxygen and nutrients to your cells. And the wall, which is called the endothelium, and we're going to talk about that term again. Endothelium is simple squamous epithelium, very thin epithelium that makes up the capillary wall. It also lines larger blood vessels, larger arteries and veins. So in the case of capillaries, because these are individual epithelial cells that make up the wall, there's a little bit of space in between each cell of the endothelium. So these cells, they actually literally squeeze out and then move into a tissue where they can reside. And some white blood cells live in tissues and where they can be attracted to tissues if there's an infection. Because during an infection, there's a lot of chemicals released, both by other white blood cells in the area and by damaged cells or dead, dying cells. There's also bacterial toxins too that can attract the white blood cells. So white blood cells are attracted by chemicals released during an infection, and that way they can migrate into that area and fight the infection. Positive chemotaxis means they are positively oriented, or they positively move toward these inflammatory chemicals that they're attracted to. And diapedesis is basically the amoeboid motion that they use. They kind of, use this amoeboid-like motion or movement to move around. They send out pseudopods that pull themselves along, and that's how they can migrate around inside the tissues. They don't have flagella or cilia to move. They have to use this diapedesis or amoeboid motion to move. Now, white blood cells, these are the what we call differential white blood cell counts that are normal. Differential white blood cell count means the number of white blood cells, or the percentage, I should say, of white blood cells in your blood. So in a normal healthy individual, these are the white blood cell counts. You can see neutrophils are the predominant white blood cell in your bloodstream. You would normally see those in a blood smear if you're looking at blood under the microscope. Lymphocytes are the next one, 20 to 25%, whereas 60 to 70% neutrophils. Monocytes, the next one, 3 to 6%, or 3 to 8%, I'm sorry. Eosinophils and basophils are the lowest in number. And as we'll see, if you look at differential white blood cell counts, it can tell you something about what's going on in the body, what kind of infection or other disease process is happening in the body. This table is just a summary of what we'll be talking about, showing the percentages as well as some of the functions, though we'll talk about a few other functions as well. But it shows some of the functions of these white blood cells. Notice that some of them, the neutrophils, eosinophils, and monocytes are phagocytic. That means they engulf other cells. They're involved in engulfing and destroying pathogens. But there's other functions as well that we'll get into. Now, there are two groups of leukocytes or white blood cells. And when you see WBC, that means white blood cell, just same thing as a leukocyte. Granulocytes or granular leukocytes is another name for them, are the neutrophils, basophils, and eosinophils. They have visible granules in the cytoplasm. In other words, when you stain them, these granules don't show up without a stain. But when you use this differential blood stain that stains the nucleus dark blue, and then it stains the cytoplasm and granules, you get different appearances based on the shape of the nucleus and the presence or absence of granules and the color of granules and things like that. So you have the granular leukocytes here, and then over here these are the agranular, meaning no granules that are visible. They might have kind of almost a little bit of a granular look, but they're considered to be agranular because there's not the obvious granular structures that you see in the cell. So here's another picture showing this. Here are the granular leukocytes or granulocytes, eosinophils, basophils, neutrophils, and then monocytes and lymphocytes are agranular without granules. You should be able to recognize from a picture which one is which, or by a description. I may put, a description of it based on nucleus shape and and the staining of the granules, or the staining of the cytoplasm, or I may show you a picture, or I may do both. And also know their functions, which is even more important. So be sure you study the picture as well as the descriptions of these white blood cells. The first one we're going to look at is the most common one, and it's a granular one. So we're going to start with the granular leukocytes or granulocytes. Neutrophils. These are make up 60 to 70% of a normal white blood cell count. And these have a very obvious appearance. They're really large, comparatively speaking. They're large cells. They have a very oddly shaped nucleus here in blue. It can have what they call 2 to 5 lobes, and that means these blobs here. That's a lobe, that's a lobe. So they can have up to 5 lobes in their nucleus, so anywhere from 2 to 5. So instead of having a nice round nucleus like most cells have, they have blobs or lobes, and they're all connected. That's where the DNA is, but it does have kind of an odd appearance. They also have kind of a pink cytoplasm with dark purplish granules. So you see a pink background, dark purple granules. So that's what you look for. And typically, if you're looking at a blood smear, these are the ones you're going to see most often. They're going to be commonly, spread out among the red blood cells. And I think at the very beginning of this lecture, I should move that picture over here. There is a picture that shows red blood cells and white blood cells, and it specifies which kind is which. It tells you which ones are neutrophils. And you want to look at that picture because it does show you, it labels the different kinds so you can see what they look like. But anyway, this is an important phagocytic cell. It is the first to move into an infected area, or one of the first to move in. It is attracted to the chemicals released during an infection. Really, those toxins that bacteria produce, or the chemicals that dead cells produce, and other chemicals that other white blood cells produce, attract neutrophils to the area. So these are really an important one for fighting infection, and if you have an active infection in your body, you'll see an elevation in neutrophil count. Now, neutrophils are like kamikaze pilots when it comes to white blood cells. They come in, they engulf the bacteria that's in the area, and they have these really strong lysozymes, which are antibacterial enzymes, and strong oxidizing agents or oxidants. An oxidant is something that oxidizes something. For example, hydrogen peroxide, I don't know if they produce hydrogen peroxide, but hydrogen peroxide is a strong oxidizing agent. And notice when you use hydrogen peroxide, you often use it to clean a wound, and it bubbles up, it starts bubbling. Well, that means it's oxidizing those bacteria and killing them. That kills the bacteria. It also kills your own cells, which is why you don't want to overuse hydrogen peroxide, because it can actually start damaging your own tissues. It's also used as a bleaching agent because of that oxidation that it does. So that's why we use, hydrogen peroxide to bleach clothes or bleach our teeth, because it does work as that oxidation also bleaches chemicals. So these neutrophils are producing similar chemicals, but the chemicals that they're producing inside the cell. They engulf the bacteria, and then they're producing these oxidizing agents and lysozymes, which not only kill the bacteria, it kills the neutrophil. So it actually commits suicide in the process of fighting infection. That's why I say they're kamikaze pilots. They kill themselves in the process of fighting the infection. So what you end up with in infected tissue is a lot of dead neutrophils that you have to get rid of. So other phagocytes will come in, not only engulfing bacteria, but also engulfing those dead neutrophils to get them out of the tissue. So neutrophils don't last for very long. They oftentimes will, they'll be produced, they might fight an infection and die in a matter of hours or days. So they usually don't have a very long lifespan because of the way they fight infections. Any questions about the neutrophils before we move on? Okay, next up are basophils. Basophils are smaller cells, They're also the lowest number, 0.5 to 1% in a normal white blood cell differential. And you can't even see the nucleus because it's got these really dark, thick blue granules. So think of basophil blue. They're blue staining, very dark, and the nucleus you can't even hardly see in there. It's kind of covered up by those granules. Now basophils are important they play an important role in causing inflammation, but they also have effects on the development of allergies. So we're going to talk about that. What they do is basophils can release chemicals such as heparin, histamine, and serotonin. They can do this in the bloodstream, but they tend to move out of the blood and move into tissues where they grow in size and become what's called a mast cell. So a mast cell is just a more specialized basophil that's living in the tissue instead of in the bloodstream. So they live in the blood, they're basophils. When they move in the tissue, they become mast cells. And mast cells and basophils produce heparin, histamine, and serotonin. These chemicals cause the inflammatory response. One of the effects is they stimulate vasodilation of vessels in the tissue, which causes the vessels to develop kind of pores in the wall where fluid leaks out. So vasodilation leads to leakage of fluid, of blood plasma, into the tissue, which causes inflammation. And inflammation here we're talking about, increased fluid in the tissue. This serves a couple of purposes. One, by having these openings in the blood vessel wall, that makes it easier for white blood cells to get out of the blood vessel and into the infected tissue. So those neutrophils and other white blood cells can get into the tissue more easily if you've got a dilated vessel. But also that excess fluid, or that, not excess, but that fluid leaking into the tissue warms the tissue. You might notice that inflamed tissue is warm to the touch. That actually increases the metabolic rate of the white blood cells so that they can more efficiently fight the infection. So you're increasing the metabolic activity of the white blood cells with that warm sensation. Also, inflammation kind of walls off the infected tissue so that it doesn't spread. So inflammation is uncomfortable and sometimes unpleasant to have, but it actually helps your body fight the infection. The problem is sometimes inflammation can get to be too much. If you have too much inflammation, it can be very painful. You can get swelling and tenderness. But also sometimes inflammation can be inappropriate. In other words, your basophils sometimes respond to non-pathogenic things as if they're a pathogen, and that creates what we call hypersensitivity reactions or allergies. For example, pollen. When you breathe in pollen, it might stimulate those basophils in your mucous membranes to produce these chemicals which cause inflammation in your sinuses, which leads to sinus congestion and nasal congestion and maybe sinus headaches and stuffiness and all that. If you have this happening in your lungs, it can lead to asthma attacks. And so basophils play a role in allergies and asthma. Hives that you might get from food allergies are a part of that too. So the various allergic reactions that we get to either chemicals that we breathe in, like pollen, or food chemicals that you might have, be allergic to, like in peanuts or strawberries or soy. A lot of people have food allergies to those things. That is an inappropriate reaction to some chemical your basophils react to, and then they start setting off an immune or an inflammatory response. And notice that one of the chemicals that they produce is histamine. Histamine, oftentimes when you have an allergic reaction, you take an antihistamine, which helps reduce inflammation, helps reduce that inflammatory response. Any questions about that? So if you had either inflammation from an infection or an allergy, then you would see an elevation in basophil counts. You'd see higher numbers of basophils than you normally would see in your bloodstream. Now, eosinophils, any questions about basophils? Okay, eosinophils, they are not as small, but a small number, 2 to 4% of your white blood cells. They have what's called a bilobed nucleus. This is the nucleus here, in blue, and it's connected by a little thread of nuclear material. That's 2 lobes, that's a bilobed nucleus. They also have very red granules. So eosinophils stain very red, lots of red granules in the cytoplasm. By the way, these granules contain chemicals. The chemicals that these cells use to fight infection or do whatever, those are in the granules. So those basophils, histamine, heparin, serotonin, they are found in these granules in the cytoplasm. So eosinophils also produce chemicals as well, those chemicals are in these granules. Now, eosinophils have several jobs. One is they fight parasitic worm infections. So if you had, say, a tapeworm infection or something like that, you would see an elevation in eosinophils in your digestive tract. They also play a role in inflammation, but they do the opposite of what basophils do. We'll talk about that as well. And they're also phagocytic, so we're going to talk about that. One thing they do is, when there's inflammation, they release chemicals that work against those inflammatory chemicals. For example, they release chemicals, enzymes that break down histamine, so they have an antihistamine effect, an anti-heparin effect, and all this other stuff. So they are involved in reducing inflammation. So they kind of help in preventing too much inflammation. Too much inflammation is a bad thing. So they help prevent too much inflammation during the inflammatory response. They also, we're going to talk about this in a second, they engulf antigen-antibody complexes. We'll talk about that in a minute. And again, they also work against certain parasitic worm infections. So they play a role in that as well. They, produce chemicals that fight these infections. Now, in terms of phagocytosis, we're going to talk more about this picture in a minute, but what we mean by an antigen-antibody complex, we're going to talk about lymphocytes in a little bit. Lymphocytes are B cell, lymphocytes are a white blood cell that produce, or B cell lymphocytes, I should say, that produce antibodies against pathogens. And these antibodies that are produced are proteins that bind to antigens on bacteria and viruses and other cells like that, or other pathogens. Viruses aren't really cells, but they do have antigens on them. Antigens can be either protein, they can be glycoprotein, they can be glycolipids, it just depends. But these antigens are different from your own cellular antigens. You have cell identity markers or antigens on your own cells, and that you should not respond, your immune system should not respond to. B cell lymphocytes, though, will respond to foreign antigens on foreign invaders like bacteria and viruses. And these antigens shown in red, that's a cell identity marker. It's telling these lymphocytes that it's a foreigner, it shouldn't be in the body. So B cells respond by producing protein antibodies that bind to specific antigens on these pathogenic organisms. And what happens is a lot of times these antibodies will cause all those bacteria or viruses to clump up, creating what we call an antigen-antibody complex. In other words, a clump of bacteria or viruses or other types of cells that then can be engulfed by phagocytic white blood cells. And eosinophils play a role in this. They are involved in engulfing antigen-antibody complexes. So this is one way of fighting infections is your B cell lymphocytes produce antibody, the antibody binds to the antigen on that bacterial cell, creating an antigen-antibody clump here in this complex, which then gets engulfed by phagocytes. This is part of your, what we call adaptive immune system that we'll talk more about later on. But this is a way that your body responds, one way your body responds to infection. So we're going to come back to this picture in this discussion when we get back to B cells, but let's go on to The next group. Any questions about eosinophils? So the eosinophil in this picture would be this guy. But also other phagocytic cells do this as well, but eosinophils do play a role in that. Now, agranular leukocytes or agranulocyte, A meaning none, they don't have the granules in the cytoplasm. Monocytes have kind of a lavender, lilac colored cytoplasm, light purplish, I guess. And they have kind of a C-shaped or kidney bean shaped nucleus. So the nucleus is fairly significant there, big C-shaped nucleus. These are very large cells, by the way. These are one of the larger of the white blood cells. Now monocytes are the second most common type of white blood cell, or no, not second, third most common type. There's more lymphocytes than there are monocytes. But monocytes are found in the bloodstream. And when they move out into tissues, they grow in size and transform into phagocytic cells called macrophages. So macrophages come from monocytes. And so there are two kinds of macrophages in your tissues. There are fixed macrophages that live in tissues all the time, especially like your connective tissues. Fixed macrophages can move around. They're not stuck in place, but they pretty much just stay in whatever connective tissue they're in. So the monocyte moves in and then just lives there in the tissue, helping to prevent infection. Wandering macrophages are these are macrophages that are attracted to an area because of an infection. So if you have an infection over here, they're going to move to that area, kind of like neutrophils. They're attracted to chemicals that are released, and they move into that area and start fighting the infection. Now, macrophages and neutrophils are important. Both of these are important in fighting infection or preventing infection. These are really important for preventing infection in the first place, but they're also important in fighting infections. Macrophages do not kill themselves in the process of fighting an infection like neutrophils do. So neutrophils are usually the first to arrive into a infected tissue, and then you got the macrophages moving in after that. Macrophages can engulf more pathogens. They also engulf dead neutrophils in the area, so they're getting rid of those dead cells as well. So they're important, kind of like the cleanup crew in an infection. But again, they don't die as quickly as neutrophils do. So you have really, several different kinds of phagocytic cells in the body. And I was going to show you, let's see, there's monocytes, but this is in the bloodstream. So monocytes are monocytes in the blood, but they change into macrophages, which are larger cells that you would find in the tissues. And there's a picture somewhere, oh, here we go. This picture here, notice that they show a monocyte, and then they show it becoming a macrophage. This would be in the tissue. Just like basophils, they don't show it here, but basophils become mast cells. And, oh, there it is. Well, they're showing it as a separate lineage there. But mast cells, now maybe there's been a change in the research. I'll have to look into it. But as I understood it, that mast cells come from basophils. But maybe they're finding there's a separate lineage there, and that's why they're showing that. Because typically, I've always learned that mast cells came from basophils. Maybe it's changed. But anyway, both of them, regardless, both produce histamines and so forth. Anyway, lymphocytes. These are very important in what's called adaptive immunity. Adaptive immunity is long-term immunity. These other cells we're looking at, they deal with infections at that moment just to kind of get rid of it. But lymphocytes are important in maintaining your immunity against organisms that you get infected with, particularly viral infections and things like that. So, if you have chickenpox as a kid, you don't get chickenpox again because you have adaptive immunity to the chickenpox virus. If you had, some other childhood disease, mumps or something like that, you wouldn't get mumps again because you have adaptive immunity to that infection. Now, some organisms we don't really develop very much adaptive immunity to, it just depends, but there are certain organisms we do develop adaptive immunity. And this is also the basis of vaccinations. Vaccinations, when we say we immunize you against something, what they're doing is stimulating adaptive immunity so that you have long-term immunity against a particular pathogenic organism that you've been immunized against. And this is very specific. You know, you can be immunized against chickenpox, you can be immunized against mumps, but a mumps vaccination is not going to immunize you against chickenpox. And chickenpox vaccines are not going to immunize you against the flu. You're going to have specific immunity developed against each specific organism. Now, there are 3 different kinds of lymphocytes. B cells, these are the ones that become plasma cells when they're activated. Plasma cells are larger cells, but they produce antibodies. And antibodies are what bind to those antigens I was talking about. We'll look at a picture of that in a minute. B cells also can become memory cells. Memory cells are the cells that carry that immunity into the future, so that if you get chickenpox, you develop B cells that become plasma cells and produce antibody against chickenpox virus, and you get rid of the chickenpox virus. Meanwhile, you've got other B cells becoming memory cells that carry the immunity or the memory of that infection, so that when you get exposed to chickenpox again later in life, your memory cells can transform into plasma cells and start producing antibody against the pathogen. So you maintain immunity. Again, every time you get exposed to chickenpox, you're going to develop more memory cells and plasma cells. And so you kind of get, you get a booster every time you get exposed to chickenpox. Or if you get the chickenpox vaccination and then you get a booster shot later on, you keep boosting that immunity. And memory cells can live for years in your immune system, or in your body, I should say. So they don't live forever, but they do live a long time. So every time you get exposed to a virus of a certain type, or every time you get immunized, you boost your immunity by boosting the number of memory cells in your bloodstream. That being said, like I said, they don't live forever. So if you were alive back in the 1950s and 60s when people were being immunized against smallpox, and you had smallpox immunity, so that if you were exposed to the smallpox virus, you wouldn't get it. By this time, unless you've been immunized again, by this time you've lost all those memory cells and you'd still be vulnerable to smallpox. So that memory only lasts for so many years before you have to get a booster shot or get re-immunized. Now, T cells, these are cells that they have a variety of cells they attack. They don't produce antibody. They have other methods of killing these things, but they also develop memory cells. They also carry immunity into the future to prevent infection in the future. They do attack virally infected cells in your body, because viruses infect your cells and then use your cells like little virus-making factories. So that, if your cells are infected with the flu virus, they'll kill those cells and get rid of them. If your cells are infected with chickenpox virus, they'll kill those cells and get rid of them that way. But they also form memory cells that maintain your immunity. Fungal infections you might get, they fight those. They also attack cancer cells. So there's a lot of research being used to look at T cells and how they might be used. In fact, they are being used to some extent in immune system or immunotherapies against certain cancers. They also attack some bacteria and transplanted cells. So if you get a tissue transplant, an organ transplant, these are the cells that can cause problems with host versus or graft versus host disease, or organ rejection, because they will attack foreign cells, including a heart that was put into your body from somebody else, or bone marrow, or other cells that are foreign to you. So they can create problems for people who get various tissue transplants. Another one, natural killer cells. These also attack many of the same things T cells attack. They were once thought not to give you long-term immunity, but now research is showing that they actually do play a role in long-term immunity. So natural killer cells may actually have some memory that they maintain into the future as well. Now here's showing a lymphocyte or a plasma cell. Let me go back to this picture of lymphocytes. And I thought I had a picture. Okay, so lymphocytes, they develop from a different cell line. Remember, they develop from, let me go back here. They develop from the lymphoid stem cell line. All these other cells develop from the myeloid stem cell line in the red bone marrow. These guys develop from the lymphoid stem cell line. All of these, though, come from a stem cell, the pluripotent hematopoietic stem cell in the bone marrow. Lymphoid stem cells give rise to T cell lymphocytes, B cell lymphocytes, and natural killer cells. And notice that here it shows B lymphocytes developing into plasma cells. B lymphocytes are smaller, plasma cells are bigger, because it's the plasma cell that's manufacturing antibody, so it needs more cytoplasm for that function. So notice there's a big change in size because now it's using its cytoplasm to make all that protein antibody. So that's why there's a change in size. Now, let me go back to the picture, wherever it is. Lymphocytes are small. They have a really giant nucleus that's kind of indented on the side or maybe slightly C-shaped. And then they have a narrow rim of cytoplasm. That's when they're inactive. When they get activated, they increase their cytoplasm. So B cells become larger plasma cells with more cytoplasm. But this is just a non-active cell here. It's not a plasma cell yet unless it gets activated by exposure to some pathogen. Now, again, they don't have granules in the cytoplasm, and again, they are important in your adaptive immunity. So adaptive immunity meaning long-term immunity. So again, you can see small amounts of cytoplasm, big giant nucleus, and then here a plasma cell, much more cytoplasm there when it's activated. Here's a picture of activated plasma B cells, and they're making antibody. Antibody, the protein has kind of a Y shape to it, and it has antigen binding sites. And here it's binding to these little, this is a pathogen of some kind. These little red things on here, if you can see those, are antigens. They're cell identity markers. They're foreign because they're on pathogens or on bacteria or viruses. And the antibody sticks to that, covering up the pathogen and again forming antigen-antibody complexes. So again, you get a bunch of those antibodies, they stick the bacteria together or the viruses together, creating this antigen-antibody complex that gets engulfed by phagocytic cells like eosinophils and macrophages and so forth. So, so that's how you get rid of a lot of infections this way. Not just by phagocytosis, but by this process of making antibody. And again, some of the B cells, instead of becoming plasma cells, some of them will become what we call memory cells. And we'll talk more about that when we get to the lymphatic system for the exam 3. But any questions about this?